Okay guys, so today I will be giving you first of the series of three presentations. It will be just general overview what spike sorting is about, how we are recording neuronal activity, and more details you will learn later. And this presentation is in something like 50% based on slides from Ken Harris. Okay, so we would like to monitor activity of large number of neurons, know which neurons fired when, know which neuron is of which type, and to estimate errors. And I will be covering all those topics. Okay, so the basic way it works is that we use tetrode, which is composed of 4 wires, and if neurons are close enough to these wires, and when neuron is firing, the change in voltage will be recorded. And the important things to know is that we are using these 4 wires to better see which neuron fire when. So for example, because this wire is closer to this neuron, spikes from this neuron will have larger amplitude on this wire, but smaller amplitude on this wire. And for this neuron, it will be vice versa. It will have larger spikes on this wire and smaller on this. So if there would be just one wire and 2 neurons would be in the same distance, it would be difficult to know if the spikes belong to this neuron or this, but having these 4 wires, it allows us to get this view at the neuronal activity from different angles, which helps with spike sorting. Okay, so this is a picture showing this tetrode wire, and you can see that usually from theoretical point of view, in this radius of something like 50 microns from which we can record signal, there should be around 100 neurons, and we also should be able to pick up signal from even larger radius of about 140 microns, and in such spherical space there should be around more than 1000 neurons from which we can record. In practice, usually we record only signal from neurons which are very close to each other and which have significant firing rates. So usually we have on one tetrode signal from 5 up to 20 neurons, which can be well discriminated with spike sorting. And it should be mentioned that this technique to use tetrode, multiple wires to record with higher fidelity signal to distinguish signal from different neurons, it was idea introduced by Bruce McNaughton in beginning of 80s. Okay, so we are using here other things than tetrodes. We are also using here silicon probes. Here is an example of silicon probe. So this probe allows us to record activity across, for example, multiple layers. And you can see that this is really great if someone would like to know how signal propagates through cortical column. And again, at 2 neighboring sites, probably there will be signal from the same neuron. Just if neuron is closer to this site, it will have higher amplitude on this contact, and here it would have smaller, and so on. And this is picture of a typical silicon probe. Silicon probes can have also different configurations. They can have multiple chunks, like up to 8 chunks, and on each chunks you have this tetrode configuration. So it is quite good because here you have this nicely equally spaced sides, so when you are recording you know from exactly which layer what's the special relations between recorded neurons. But the disadvantage is, as comparing to tetrods, is that here you cannot move each chunk separately. If you are moving, you have to move all chunks together. With tetrod, you can move each tetrod separately. The one other point is that these electrophysical recordings are taking a lot of hard drive space. 1 hour recording from 64 channels is approximately about 20 gigabytes, and if someone records for let's say 24 hours, these files can get really huge. So for that we not only need good amplifiers and those probes, but also we need a good system for storage, for storing this data. Okay, so here is picture of a neuron, and there was few experiments in Yuri Buzaki lab where they put electrode intracellularly and they also put other electrode silicon probe extracellularly, and they could measure how the activity in neuron, these intracellular spikes, relates to what we record extracellularly. And here is example that this is a shape of intracellular spike. So when neuron is sending signal, it's generate this spike, and if electrode is inside neuron, you will see this type of shape. Extracellularly, it will be similar to minus derivative of this intracellular signal. And this is related to the direction in which ion flows, but exact information about it is it would require another topic, another lecture. Okay, so if we have this neuron, we can lower silicon probe or tetrod, and one thing to know is that the shape of spikes will depend where is exactly position of this electrode in relations to cell body. So if we have contact close to cell body of this big pyramidal neuron, then we will see these nice big spikes. If we have site which is closer to this apical dendrite, then the same spikes will have this decreasing amplitude if, for example, neuron is bursting. So here I'm talking about one specific case when pyramidal cells sometimes can fire this multiple spikes in very short distance from each other, so it is less than 3 milliseconds interval between spikes. And in this case, here in this apical dendrite, there is not enough time for this potential to build up, to recover from each spike, and for that you see these smaller spikes. And later I will talk about it, how it influences spike sorting. If we have some electric contact which is closer to dendrites, then we still will be able to record spikes, but the quality of the spikes and shape and size will be different. So okay, here is another illustration showing where how position of recording sites in relations to neuron, how it will affect what we see on our recording. And sometimes those waveforms can be really have these bizarre shapes when we put electrodes closer to dendrites than to cell body. And some people spend a substantial amount of time trying to understand it. And there are some models which are describing how signal propagates through dendritic tree and how it is affecting shape of action potential. So what you can see here is that here in the center it was cell body, and those waveforms shows what would be the shape of spike at given distance from cell body. And you can see that it is not just a scaled version. This shape is also the shape of the spike is also changing. So this is important point for spike sorting, is that 90% of time you will see these nice spikes on your recordings, but sometimes you will see some really strange waveforms. So you should be really suspicious and be careful if this is not some artifact. But you know, biology is strange, and sometimes such strange spikes are completely good spikes to be included in analysis. Okay, so here I'm showing you data recorded from silicon probe from 8 chunks. Each chunk is this different color, and on each chunks we had this 8 different recording sites, and this is something like 100 milliseconds of data. And you can see that when there is no neuron spiking next to electrode, you see this kind of relatively smooth trace, but sometimes you see this big jumps in voltage, and it is when neurons are firing. One thing to notice is that those, for example, spikes are different from these spikes. It means that there was some neuron which was probably closer to this electrodes, but quite far from this. But these spikes are probably coming from another neuron which was in more or less equal distance from all those electrodes. So this is the idea about this spike sorting, that we will record this continuous voltage, and later, looking how the shape and the amplitude of spikes changes across different channels, we can say that those spikes probably belong to one neuron, those to different, and so on. Here is 1 second of data, and it is to demonstrate that when we record there is this a lot of slow oscillatory activity. So this is usually reflecting global input to that area, and these continuous traces are called local field potential. And it's difficult to notice, but if you look closer you will start seeing that there are some small jumps in voltage, and those jumps are spikes. So now the question is how to detect those spikes when there are those big oscillations. And the answer to that is to do filtering. So here is replotted data from previous slide, and here is after filtering. So after filtering you start seeing that there are some very clear jumps in those traces, and those jumps correspond to times when we saw before those spikes. And you can start seeing that certain spikes are very similar across electrode, like this first spike is similar to the second and is also similar to that, but there are also other spikes which are much smaller. And again, this is what we will be looking at spike sorting. We will try to see what are the groups of similar spikes, so we can say that this spike and this spike and this spike is coming probably from cell 1, and those smaller are coming from cell 2. Okay, so what I told you is that local field potential, it is this continuous signal which is primary at low frequencies. After filtering we are getting only spikes after this high pass filtering, and because spikes have this much higher frequency oscillations. And here is example of recording from one of our system here. And in our system you can record 2 types of data. You can record this continuous local field potentials, and here is example of 4 traces from one tetrad. And you can see that there is spike on this channel. It is also on other channels. It means it was a nice cell which was close to all the tetrads. But what our system is also allowing us is that it will do this online filtering, and it will detect when those spikes occur, and what will be saved is only time of the spike and waveform of the spike. So in one mode you can record all the signal. In second type of data files you can have only times of spikes with this shape. So the advantage of having everything is that there is a lot of useful information in local field potential. The advantage of having only spikes is when you are recording for long times, like recording for 24 or 48 hours. The data, if you would record continuously, it would be just data files would be too large to handle. For that, for such long recordings, people usually record only spikes. Those two modes are not exclusive. You can record both local field potential and also times of spikes with waveforms. And as you can see, here is the waveform of this spike at these 4 different channels, and again you see that it has the highest amplitude at one channel. It means it was probably closest to this channel. Okay, so to summarize this part, I showed you that if we stick electrode somewhere close to neurons, and if we amplify the signal and filter it, we will see spikes, and after filtering it is easy to detect them just by thresholding. After detecting each spike, if we zoom in, we will see that each spike has spikes from different neurons have different amplitude usually, but also different shape, and spike sorting is basically this job of clustering this data and saying that this type of shapes are probably from one neuron and this longer waveforms are probably from other neuron. And this is waveform of those two different waveforms overlay on each other to show you that they are quite different. And what people did previously is that they were trying to do spike sorting. They were overlaying those waveforms on each other and were saying that, oh, if some waveform is passing through this point and is also passing through this point, it will be spikes from one neuron. If some waveform is passing through this point and through this point, then it will be spikes from second neuron. And this is what people call this manual spike sorting based on whole waveforms. But there are a little bit we can do a little bit better by reducing waveforms to only specific features. So for example, we can create some feature vector, and for that there is really good principal component analysis, which will help us to only look at most important features of those spikes instead of on entire waveforms. And again, just a reminder to group that if something is not clear, either with slides or with my accent, stop me and I will be glad to explain. Okay, so here is example of let's say 10-15 milliseconds of data. You can see here example of 3 spikes, and here are probably spikes from 2 different neurons, and if you collect data from let's say 20 minutes of recording, you will have many waveforms, and if recording was nice, you will start seeing that in this case there are those two clear clusters. So again, the typical approach it would be to trying to find that these waveforms are going through this point and to separate in this way. But the other way is to use this principal component analysis. So this method is looking at which point data is the most variable, where are the biggest difference between those waveforms, and it automatically can map it in this principal component space. So you can think about it like looking at amplitude and some combinations of spike waveforms like width. So usually, because the usually highest variability is at the peak of spike, so first principal component is pretty well correlated with amplitude of spike. So you can think as an amplitude of spike. And second principal component is, let's say, maybe telling what is the amplitude here of this hyperpolarization period. So now, instead of having this, let's say, 30 or 60 points describing waveform, now each of those spikes is described only by two numbers: first principal component and second principal component. If you think about it like that, each waveform is now described by approximately by amplitude of spike here and amplitude of spike here. So now you can see those two clear clusters, and now it is really easy to draw some border between those clusters and say that all those spikes here belongs to one neuron, and those spikes here belongs to second neuron. So again, each dot in this plot, it is one spike, and on this axis you can think as amplitude of this part of spike, on this axis amplitude of this part of spike. So if spikes have different shapes, you would start seeing those that they fall in different places in this space. So for spike sorting, it's not so crucial to understand exactly what principal component analysis does, but if someone would like to do some data analysis later, it's really useful to know a little bit linear algebra and this method. Here is another example where we have an example of two neurons, neuron 1 with this waveforms, neuron 2 with those waveforms, and here, because we are using tetrodes, so this is average waveform from one channel, second, third and fourth. And you can see that here is neuron with this cloud representing one neuron. So this neuron probably has low amplitude spikes, and here are two neurons with pretty similar amplitudes at channel 1. So at channel 1, at channel 2 and channel 3. So in this case, these points overlap between neuron 1 and neuron 2. But if we look at second principal component, which is looking not only at amplitude but also what's going on after this initial peak, then you can start seeing that here in this view, looking at channel 3 principal component, second principal component, you can start seeing clear differences between this red and between green cluster. So here green and red were overlapping, but looking at this, looking from other angle, you can start seeing that those are two separate clusters. And for humans, it's a little bit difficult to do this because we can only see on monitors 2 dimensions. For that, I will be telling you a little bit how computers are doing it. That in computer, computer can easily look in 100 dimensions, so he can do some pre-clustering for us, and I will be talking about it. So I told you that most often people are using this principal component analysis, but also here we are using in addition something what is called energy. Energy you can think as a area of spike. So if spike is bigger and has this larger area, it will have a larger energy. And here example of multiple spikes, multiple average waveforms recorded at 1 tetrod, and you can see that here it would be really difficult with this typical old method to find what are the differences between those spikes. But plotting in this energy space, you start seeing those clusters, those places where there is higher probability for those dots to be located. And again, each dot it is describing one spike, and you can and this is you can think about it as energy 1. It is what was the area of that spike at electrode 1, and here what was the area of the spike on electrode number 4. So for example, you see that here on this electrode number 1 the green spikes are very prominent, and you can see that on this axis, reflecting energy on this first electrode, you see this green cluster here, and for example on the fourth electrode the highest amplitude and energy were for those brown and green spikes, and you see those green and brown clusters. Okay, so it is just illustration that looking at not only at the whole waveforms but also at features of spikes as expressed by principal components or energy can really help us to see that different spikes, different neurons tends to have spikes with different shapes. Okay, so usually after recording we will after using this principal component analysis or energy, we will see those multiple clouds, and the spike sorting or clustering, it is just to try to find what are the borders between those clusters. And again, for human, it's a little bit difficult to say where exactly this border between those two clusters should be. For computers, it's a little bit easier because there are good ways to evaluate what is the density of points and where should be the best dividing plane. Okay, so we can do it manually. By manually, I mean to manually say what is the boundary of each cluster. The other way is to ask computer to do it purely automatically. The problem is that there are a lot of special cases for which it's difficult to train computer to do it correctly. So for that, the best model so far is to do a hybrid model where computer will do initial clustering and humans later will correct and see if how correct computer's job. So it is a really good type saver and also improves quality. And this is how we are doing this here. So we are using program called ClustaQuick, which is doing for us this automatic clustering. Here is, for example, data from one tetrad, and you see that this program assigns different colors to different clusters, and usually this program is over clustering, because later for humans it's easier to say that two clusters belong to the same cluster than to find borders between clusters. So it's easier to merge than to divide. So this is example. Later, with some more careful analysis, and this will be telling you, Masami and David, what are the rules to infer that, for example, in this case, those two clusters were belonging to one neuron, and also so those three clusters, green, blue, and red, were merged into one cluster. And there are many possible ways in which computer can introduce some errors, and this is this job which human has to do during spike sorting to correct this usually over clustering. In this case, what was the problem? The problem was that, as I told you, some neurons sometimes show this burst, with many spikes which occur in very short distance from each other, and usually the first spike in burst has the highest amplitude, and following spikes have a little bit lower amplitude. So if you plot this in this space of principal components or energy, which is largely reflecting amplitude and shape, you will see that this cluster will be very elongated. So here probably it will be spikes from the beginning of this burst with this highest amplitude, and spikes which are far later will have this lower amplitude. And it is really non-trivial problem, because you will see that those spikes these spikes are quite different from those spikes, but nevertheless it will be your job to accurately say that, oh, these spikes are really belonging to this. And here is just a picture showing how on average this amplitude of spikes and shape also changes in burst. What was the criteria here to figure out that those spikes are belonging to the same neuron? It was to look at cross-corelograms. So cross-corelograms are telling you what's the probability of one neuron firing of spikes from one clusters when neurons from other clusters are firing. So, for example, this cross-corelogram, what is telling is that when we had spikes from one cluster, the spikes from second clusters tend to be the highest probability here, here, and here, and it is at those very distinct bands. So it means that this usually can only happen if this neuron is bursting, that the second cluster can fire with such regularity in relation to the first cluster only if it is belonging to to the same neuron. So this regular burst in this cross-corelogram are reflecting that usually the time of this second spike is very much reproducible across bursts. So if it was in first burst something like 2.8 milliseconds, it will be almost 2.8 milliseconds at the second burst, and so on. So it is one of possible problems which we have to be aware when doing spike sorting, and computers are pretty bad to figure out that this is from one neuron. Okay, the second problem is drift. So here what I'm showing is that on this axis I'm showing what is the energy of spikes at electrode 1, and here I'm showing how it changes in time, and here it is something like 2 hours of recording. And what you can see is that this energy, this area of those spikes were pretty stable here, and was decreasing later, and here it was kind of messy. And here each color is indicating different clusters, so spikes from different neurons. Now to identify here which spikes belongs to this green cluster, which spikes belong to this cluster, is a pretty difficult job, and nobody expects you to do it with 100% accuracy, but because it's difficult and sometimes impossible, but try your best, try to prove that you are better than a computer. And sometimes there are big problems when we have big drifts, and it is another example, unfortunately from one of my experiments, where you can see again some 2 hours of recordings, and on the beginning everything was pretty okay, but later something happened, and now you have big variability in spike waveforms, and it's difficult to say. Maybe it happens that suddenly completely different groups of neurons are firing here as comparing to here. So yeah, it can happen that you have completely different set of neurons active here than here. But the more likely explanation is that something happened during experiment, that when we insert probe, then sometimes brain is bouncing back, and for that, after inserting electrodes, usually with this acute recordings, we are waiting for something like 30 minutes before starting real recording, so we can give time for brain to bounce back from this pressure which electrodes put on it. But you know, sometimes brain, I don't know, maybe some something moves a little bit in relation to electrodes, and now we are recording from completely different set of neurons. So even by shifting by 20 microns, it would cause that now shapes of neurons are quite different as it was before. And again, this is something what is a very difficult problem, and yeah, Masami and David will be telling you more how to deal with such ugly situations. Okay, so one other thing I would like to tell you about is that yeah, we will do this spike sorting or clustering, and the question is how good job we did with that. And there are a few measures which can help us with that. And so for example, here we have 3 clusters, and the simplest way to say how good we did with clustering is to see if, for example, center of this cluster, how close it is to center of other clusters. So if this cluster is well separated, well differentiated from other clusters, if it has high distance, it means that it is we are pretty sure that spikes here are belonging just to this neuron and not to other neurons. But what we saw is that most clusters are not spherical, they are rather elongated, and for that we are not using just a simple distance to measure what is the distance between center of these clusters to other clusters, but we are using distance which is normalized by variance. So it means if cluster is spreading mostly in this direction, the variance here is larger, in this direction variance is smaller. So it means that if we have point here, then the distance from the center, it will be quite large, because it will be not along the highest variability. But if point would be here, it would be very close to the center of this cluster in this using this Mahalanobis distance, this variance normalized distance, because cluster spreads in this direction. So if there would be some other cluster here, it would be high chance that we could misclassify spikes. But because this cluster is here and some other clusters are here, it seems like this cluster has pretty good separations from other clusters. I have MATLAB code to do it. It is not a standard thing in this group to apply this to all recordings. So if someone has some MATLAB skills and would like to implement it in some nice GUI, I can help with that. Neurons can be automatically quantified if they were well isolated from others or not. Another way to classify if neurons are well isolated or not, it is giving some scores by eye how well it is isolated, and Masami will be telling about his scheme to approach it. But to do it automatically, it's relatively straightforward to implement this Mahalanobis distance. And here is some quantifications, what's the meaning of this isolation distance. So if there is a small isolation distance, if two clusters are close to each other, it means we have a high probability of incorrectly including spikes from other neurons. If there is a high isolation distance, it means that probably we had this low probability. So it is important when we are doing some analysis, and it is crucial for us to know that those neurons, those spikes were really from single neuron and not from group of neurons. In this case, it's really critical to look only at neurons with this high isolation distance. In other cases where we are less concerned with activity of one neuron and we are okay with clusters being a little bit contaminated, this type of analysis is less important. What else can we learn from spike waveforms? It's also good to remember that most interneurons have different spike waveforms than pyramidal cells. Pyramidal cells have these big cell bodies, and usually spikes are broad. So here is example of this blue of pyramidal cells with these broad spikes. Interneurons have these smaller cell bodies, and most interneurons have this much shorter spikes. So if we look at what's the width, half width of spikes, so we look what is the width of spikes at the half amplitude, and second we can look what is the distance between minimum of spikes and this hyperpolarization. So if we do it, we can see that usually neurons, these points, cluster in two different groups. And here usually it is putative interneurons. This means putative pyramidal cells. So it's really useful information to know, because in brain it's very important to know what inhibitory neurons are doing, because they seems to be this like police officers which are controlling traffic, how signal is propagating. So to know which spikes are coming from, probably from interneurons, and which are coming from pyramidal cells, can be really important for certain types of analysis. In addition to looking at spike width, looking at cross-chirograms also helps to identify interneurons, that if there is some neuron which is if we look at cross-chirograms between two neurons and we see this shape, which means that one neuron fired much less, the probability was reduced almost to zero right after firing of other neuron, it means that this neuron was receiving a lot of inhibition, And if this inhibition with this short latency after this first neuron, it indicates that it's probably monosynaptic inhibition. Question? No. Okay. Good. So the question is, what would be the short latency? Short latency, it means below 5 milliseconds. This is what you expect for monosynaptic connection. And on all those things, there is a lot of literature. So if you are interested, there are places to look for. The other thing what we can learn from spikes is also what is the spatial distribution, especially when you are using silicon probes which have this very well defined distance between recording sites. Then if you see that some neurons has the highest spike amplitude at this electrode and others neuron has the highest amplitude at this electrode, you can think with quite good accuracy that this neuron was somewhere here and this other neuron was somewhere here. So you will know not only activity of those neurons, but also you will know how in space they were, how far they were from each other. And in this case, it is taken from Barto, from Buzaki lab. They identify based on waveforms which neurons are interneurons, and interneurons are those circles, and pyramidal cells are triangles. So here they could make this nice network. What is the connectivity between pyramidal cells and interneurons, and how it is changing in space. Okay, so I'm done, and now is time for questions. Was anything not clear? It means I did excellent job. Thank you, guys.